Hi ladies, this is chapter 15 on regulation of gene expression. What I want you to be thinking about as we're going through this presentation, if we have the same DNA in all of our cells, why do we have different cell types? So if you think about it, we have neurons, we have bone cells, we have red blood cells, white blood cells, skin cells, liver cells, lung cells, all different types of cells that all look completely different. Why is it that we have all these different cells, and how did they get to this way that they were so different? So what we're going to be talking about today is the regulation of gene expression. We have differential gene expression in all the different cells of our body. This means that at some point during our development, we had cell differentiation take place. So this is the process by which cells become specialized in structure and function. We're going to do a little bit more with this next time we meet. Any abnormalities in gene expression are usually a result of a mutation. So you just watched a video on mutations, and these mutations can lead to different diseases, possibly such as cancer. Okay, so let's look at how gene regulation happens. So gene regulation can happen at a variety of different places. I've introduced a couple of these, but I'm going to go into a little bit more specifics at the transcription level. So the first thing is that we haven't talked about yet is chromatin modification. So remember that chromatin is your DNA with little histone proteins that kind of keep it all wound up and protected. So we're going to look at chromatin modification, transcription, RNA processing, and then translation, and then degradation of mRNA and protein. So the first thing I want to talk about is what we usually refer to as epigenetics. So this has to do with chromatin modification. So two different things can happen. One is methylation, and the other is acetylation. So with methylation, it's going to cause the chromatin to tighten up, so to get wound really tightly around all those histone proteins. With acetylation, so you can see here, this is pretty tightly wrapped. With acetylation, what's going to happen is acetyl groups are going to be actually added to these little histone tails, and it's going to actually cause the DNA to become a little looser. When the DNA that chromatin is a little less tightly packed, our RNA polymerase can get in there and it can increase transcription of those genes. Whenever the DNA is wrapped really tightly, transcription is essentially going to stop on those genes. So this is the first way that cells can become a little bit different. The next thing we're going to look at is an operon model. So this is a very simplified version of what happens in eukaryotic organisms. Eukaryotic organisms are very complicated. We're going to be focusing on operons that are in bacteria, and they're very simple in comparison. On eukaryotic organisms, sometimes the genes for regulation are on different chromosomes completely. With bacteria, because they have a single chromosome, that circular chromosome, it's a little bit simpler to regulate. So let's first talk about what an operon is. So basically, you can think about an operon kind of like a light switch. It's like an on-off switch for a gene. So the operon is a stretch of DNA. Okay, it'll include the operator, the promoter, and any of the genes that they control. So the operator is essentially the switch, so the on-off switch. Okay, it's going to turn the gene on, it's going to turn the gene off. The promoter, if you remember from the transcription lesson, is where the RNA polymerase has to be able to bind and start transcription. Okay, then of course the gene that needs to be transcribed. So with our operator, the way that the operator can be turned off essentially is with a repressor. So this is the way that it can be turned off. Sometimes repressors can also be turned off, so you can turn on the gene by turning off the repressor essentially. And I'll show you a picture of this in a second, how this works within an inducer. So the first example I'm going to introduce is the trip operon. So this is the production of tryptophan. So you can see here you have your precursor, enzyme 1, enzyme 2, enzyme 3, and this is going to make tryptophan. So when you have too much production of tryptophan, it is actually going to inhibit enzyme 1 and stop the production of tryptophan. When you have too much of it, there's no reason to keep making more of it. So this is what we call feedback inhibition. So this is a negative feedback pathway. Here is what the operon looks like. Remember, here's the promoter where RNA polymerase has to bind. Here is the operon, the operator, the whole thing's the operon. So here's the trip operon, including the genes, the promoter, and the operator. The operator is the on-off switch. Okay, at a different part in the DNA, usually fairly close in bacteria, you have your stretch of DNA that's actually going to code for the repressor. So this is the regulatory gene, okay, which will be transcribed to make mRNA, which will make a protein. In the case of the trip operon, okay, the repressor is typically inactive. Okay, it'll only become activated with the help of a co-repressor, which in this case is tryptophan. So when there's a lot of tryptophan around, it'll bind to the repressor, and then in this case, it'll cause the repressor to become active. The repressor can bind to the operator, turning off the gene transcription. Basically, it blocks RNA polymerase from being able to proceed from being able to start at the promoter and transcribe the gene. Okay, you'll normally see repressible enzymes. Repressible enzymes are usually functioning in anabolic pathways. So in this case, we are actually making something. So when you make too much of it, you can use a co-repressor such as tryptophan, which is what's being made by the pathway, to actually come back in and repress the pathway and stop the production of tryptophan. So if you already have too much of it, there's no reason to make more. Okay. Kind of a little bit of an opposite example here. We have inducible pathways. So this is an inducible. So inducible enzymes are usually found in catabolic pathways. So this would be a pathway that usually makes like an enzyme or something like that to possibly break something down. So in this case, you have a regulatory gene, and this regulatory gene is going to allow for the production of mRNA, which is going to make a protein, and this is going to be a repressor. And when this protein is made, it's an active repressor, so it can instantly bond to the operator, that on-off switch, and it can turn and not allow RNA polymerase to transcribe the gene. Okay, so this is what's happened. This is what occurs normally. So the lac operon is important because it helps to break down lactose sugar. So it's good that it's initially turned off, because if there's no lactose, there's no reason to make the lactase enzyme in order to break down lactose. So it's usually turned off unless you actually have lactose present. In this case, the presence of lactose, okay, this is what we call an inducer. Okay, the inducer will bind to the active repressor, and it will actually inactivate the repressor, removing it from the operator and allowing RNA polymerase to make lactase, which will allow this individual, well, in this case bacteria, to actually break down the lactose sugar and use it for energy. So the presence of lactose is going to turn on this gene, okay, by inactivating the repressor. Transcription factors are also very important with the transcription of genes, so they're required. But What also may be needed are enhancers. So enhancers are basically control elements for a gene, and usually they're located a little farther away from the intron, so the gene that's actually being transcribed. And enhancers require activators. So I'll show you how this works. So you can see here's that promoter that we were just talking about. Here's your gene of interest. And then farther down, you have these distal control elements that we call enhancers. Okay, when an activator is present, it will turn on the enhancer, which will help with the binding of transcription factors and RNA polymerase to the promoter region. Okay, will help with polymerase binding to the promoter region, will help with all these transcription factors binding to the promoter region to kind of initiate the transcription of this gene. So those are enhancers. Okay, so let's go back for a second. So we did chromatin modification, transcription. In the previous presentation on translation, I also included a section on RNA processing. So remember, alternative RNA splicing is really important. You can make a lot of different combinations of mRNA, which will make a lot of different proteins based on how the introns and exons are spliced together. And the last thing I want to show you is a really great example of how once mRNA is made, or even with protein, how there's different methods that can turn off the production of a protein just by removing the mRNA that's produced, or even if you produce a protein, okay, destroying the protein will in fact also destroy whatever effect that protein has. So one of the phenomena of inhibition of gene expression is what we call RNA interference, or RNAi. And RNAi usually involves the destruction of mRNA. So you can see here, if you have an mRNA that's produced from transcription, if it's not needed for expression, okay, or if there's too much mRNA, or if it's been around for a while, a lot of times this mRNA needs to be destroyed. And destroying it with RNAi, this RNA interference, will essentially remove the mRNA, so then the protein can't be made. I am going to include a little video on Haiku that we will watch next week to kind of just show you how RNAi can work a little bit more in detail. But for right now, all you need to know is that RNA interference will typically destroy mRNA and can also help with the regulation of gene expression. Please bring any questions with you next week. We will be reviewing these topics, and have a nice day.